Welcome dear students. In this video, we are going to learn about G protein coupled receptor signaling. This G protein coupled receptor signaling pathway can be divided into 5 categories. First is Muscainic acetylcholine receptor pathway. This receptor is present in a muscles called heart muscle. I will discuss about this all different category of G protein coupled receptor in more details. The second type of this G protein coupled receptor can be called rhodopsin. That is, this rhodopsin is actually G protein coupled receptor that is present in rod cell of our eyes, rod cells. Okay. Another way it can activate, that is called by activating adenylyl cyclase enzyme. I have mentioned in my basic concept class. Then this adenylyl cyclase activate or produce CAMP, that means cyclic adenosine monophosphate. This CAMP activate another protein called protein kinase A, and this thing actually performed by beta 1 and beta 2 adrenergic hormone, or you can say adrenergic receptor, that means epinephrine or norepinephrine receptor through this pathway, or glucagon hormone, or you can say serotonin neurotransmitter and vasopressin hormone can do this pathway through GPCR signaling pathway. In case of rhodopsin, the initial point is light photon activates rhodopsins, and in each case they activate different molecules. Like in case of this beta 2 beta 1 adrenergic receptor, where these receptors are present? Beta 1 present in heart muscles. You can see here muscarinic acetylcholine receptor is also present in heart muscles, but here it is a called pacemaker cell. Different locations. Here beta 1 is heart muscles. Now beta 2 present in different locations, like it could be present in the liver cell, adipocyte means fat cells, or it can be present in the bronchial smooth muscle. So these two receptors can present in different location, and when this beta 1 and beta 2 epinephrine or adrenaline hormone binds with these cells with the receptor, they actually activate this process, adenylyl cyclase, CAMP and PK. Also glucagon, serotonin, vasopressin go through this pathway. But when light falls in the rhodopsin receptor, because rhodopsin is actually a receptor, G protein coupled receptor, when it falls, they activate different effector protein, because this is the effector protein. This is example of effector protein. But when rhodopsin activated by the photon, they activate another effector protein called CGMP phosphodiesterase enzyme, that actually degrade the CGMP and makes GMP, because we know phosphodiesterase actually degrade CGMP, cyclic GMP. So you can see here they perform different function. Like in heart muscles, pacemaker cells, the acetylcholine, actually the ligand, actually inhibits some G alpha proteins. Actually this G alpha is called G alpha. I will actually show you this all thing, but I am just actually mentioning here the overview of this molecule. This G alpha actually inhibit the adenylyl cyclase enzyme. In short, I am writing is adenylyl cyclase. So it inhibits adenylyl cyclase. So you can see muscarinic acid pathway, the effector is actually inhibited by the G alpha. But in case of this pathway, another G alpha, G protein is present called G alpha S. S means stimulatory and I means inhibitory. So there are different type of G protein present. This can activate adenylyl cyclase, but in this case G alpha inhibit acetylcholine, but G beta and gamma performs the activity role for opening of potassium channel in muscarinic acid or muscles, heart muscles. I will show you in more details, but just I am giving the overview. The fourth pathway of G protein coupled receptor is like alpha 1 adrenergic receptor. So this can be the effector protein is here PLC, means phospholipase C beta, that actually cleave a phospholipid called PIP2. PI means phosphoinositide 4,5 bisphosphate. So it cleaves this product and produce IP3 and DAG, inositol triphosphate and diacylglycerol. These two are second messengers. These are the second messenger. They activate another protein kinase, is called PKC. You can remember C for C and A for A. Okay, this is fourth type of activation. This type of activation occur in alpha 1 adrenergic cells. Alpha 1. In case of alpha 1, this process occur. What happens here? Alpha 1 receptor, adrenergic receptor, present in cells like smooth muscle cells of intestinal lining or intestinal tract, in case of skin and kidney cells. So this actually receptors present in these cells, when this alpha 1 adrenergic hormone or epinephrine actually bind with this receptor, they activate a different signaling. You can see here the effector is different. So everywhere this effector is different. I will show you in more details in next video. Another five term last mode of activation is in case of it is also the same, that PLC beta is activated and produce this IP3 and DAG, but the ultimate result is production of nitric oxide. In case of this type of signaling occurs through this type of signaling. Yes, I want to mention here that this protein that actually helps in the production of alpha 1 adrenergic receptor, the G protein is called G alpha Q. G alpha Q. Here is S, here is I, here is Q. And in case of nitric oxide producing cells, we call the G protein called G alpha O. And this actually activate these proteins. But here you can see it inhibits the effector protein. Now these cells that produce nitric oxide, they actually so G alpha O is present in the endothelial cells. You can write here O, endothelial cells, and this is actually a receptor called acetylcholine receptor present in the endothelial cells. When acetylcholine bind with this receptor present in the endothelial cells, they produce nitric oxide through this GPCR process. So you can see here different thing occurs. In case of muscarinic acid, potassium channel is activated, but in case of rhodopsin, PDE activated and GMP is produced. In case of adenine cyclase or G alpha S, in case of beta 1 or beta 2 adrenergic receptor, PK is the last product. In case of alpha 1 adrenergic receptor, the last product is PKC. In case of endothelial cells, the last product is nitric oxide. So they produce different type of molecules by the same signaling pathway. So in this class, I will give you the basic concept about G protein coupled receptor, how it actually works in all this system. But in my subsequent classes, I will discuss about each and every topic or pathway in more details. So the fact is, let's suppose this is the plasma membrane and this is a G protein. I have mentioned in my previous class that G protein has an N-terminal side extracellular domain, then 7 transmembrane domain. So you can see here 1, 2, 3, 4, 5, 6, 7. So this is the N-terminal and C-terminal. So G protein coupled receptor is actually a protein that passes the membrane 7 times. So this is called 7 transmembrane domain. This is the N terminus and C terminus present in the cytosol domain. Now, this G protein, when do not bind with this ligand, like it could be epinephrine or adrenaline hormone, glucagon, serotonin, vasopressin. You can see here other examples like acetylcholine. When this receptor do not bind with any proteins, then one thing occurs, that is the G protein, monomeric G protein that present in the plasma membrane have 3 subunits that is associated with G protein coupled receptor. This is the receptor, G protein coupled receptor. Coupled means it is associated with a trimeric protein. Trimeric. Why I am called trimeric? Because this trimeric protein have G alpha subunit associated with another 2 subunit called beta and gamma that is actually togetherly associated. They do not actually differentiate. So if I write this G alpha, then there are 2 other subunits called beta and gamma. This G alpha is actually associated with or ligated with the plasma membrane directly, and in the gamma subunit also bind with the plasma membrane, not the beta. Okay. And another thing is this G alpha protein interact with the gamma protein, not with the beta protein. Okay. Now, one thing occur when there is no ligand. When there is no ligand bind with this receptor, this trimeric protein remains togetherly. Okay, they remains togetherly. But when ligand bind with this receptor, a structural change occur in the G protein coupled receptor that helps in the association with this G protein, G alpha protein. You must remember G alpha protein. This G alpha protein associate with this G protein coupled receptor. I have mentioned in my basic concept class that G protein can be on or off state. When a G protein associate with GDP, it is off in state. When a G protein associate with GTP, it is on in state. When there is no ligand, this GTP is G alpha protein is associated with GDP. In that situation, they actually associate with the beta gamma subunit. But when the ligand bind with the G protein coupled receptor, a structural change occurs in the G protein coupled receptor so that this G alpha protein can bind with this receptor. So now G alpha protein will actually move from this beta gamma portion to this receptor, bind with this receptor. Another term I have mentioned in my basic concept class, that the transformation from G alpha to GTP, G protein GDP form to GTP form helps by a protein called GAF, guanine nucleotide exchange factor. Some molecules that can actually exchange the GTP with the GDP with the GTP. This G protein coupled receptor acts as a GAF that can exchange the GDP with the GTP. So it can bind with GTP. And when G alpha protein binds with GTP, it is actually active in nature. So the protein becomes activated. Okay. Now this protein can activate another protein. This protein actually activate the effector protein. So this beta gamma portion remains, but in some cases, like in case of muscarinic acetylcholine receptor, G beta perform the main activating function, but the G alpha protein perform the inhibitory function, which is called G alpha I. There are different type of G alpha proteins present. So GTP association is very important, and GPCR, G protein coupled receptor, actually function as a GAF. So I have mentioned that if this G protein remains as a GTP condition, this could be harmful for our cells for a certain period or certain time. So this GTP should be converted into back to the GDP in off state, and this is performed by another molecule called GAP, that means GTPase activating protein. This G alpha protein is actually has an intrinsic property to destroy this GTP to GDP, so it can become inactive. So this is the mode by which it actually works. So in case of GPCR pathway, this GPCR receptor is the GAF and the G alpha protein has the GAP property. GTPase activating property. And sometimes it actually occurs that the hydrolysis timing of this G alpha protein to break down this GTP to GDP is less. So another gap protein actually facilitated this hydrolyzation, so that the G alpha protein GTP is actually faster, break down faster. I'm telling you another time, the GTPase activity, I'm mentioned here that the GAF is the GPCR, and the G alpha protein has an intrinsic property. It can destroy the GTP to GDP by itself, but the rate of destruction or the hydrolysis is very low. To destroy the GTP to GDP, it actually can take the help from other gap protein we call RGS, regulatory G signaling protein. Okay, so in this class we have mentioned that there are different types of signaling present. The basic thing is that when the receptor not bind with ligand, it is associated with this beta gamma. When ligand bind, this G alpha protein actually associated with the GPCR. GPCR acts as a GAF. It removes the GDP and make it GTP, and it becomes activated. Now this protein can activate effector protein. Like the effector protein could be adenylyl cyclase that can convert ATP to CAMP. Now CAMP can activate pKa. How it activates, I will show you in more details. This G alpha protein can activate another protein, you can say effector protein like PLC, phospholipase C. Let's suppose PLC. This PLC then break down the PIP2 into IP3 and DAG and activate PKC. So that's why this process actually occurs. So this G alpha protein is very important, that actually activate the effector protein, and the effector protein then activates other protein, the ultimate protein. Now I will show you here some different G protein that functions, and in my different G protein coupled receptor classes I will describe this all mechanism in more details, because in one video I cannot complete all this process in more details. I will only describe about this signaling process in this class. The different type of G alpha proteins are G alpha S, G alpha I, G alpha T, G alpha Q, G alpha O. Another is G alpha olfactory. This G alpha S means stimulatory. That this G protein associated with the G protein coupled receptor can activate the effector protein, that is called the adenylyl cyclase, and it actually make the second messenger. I have mentioned in my previous basic concept class, second messenger like CAMP. It actually produce more CAMP. And how it is activated? Receptor. These receptors are this beta 1 and beta 2 receptors. So this is the all receptors that are actually activated by this G alpha S. You can see beta 1, beta 2, glucagon, serotonin and vasopressin. I have mentioned here adenylyl cyclase, CMP and PK. Here this G alpha stimulatory protein work. You can see here. G alpha I actually activate the CGMP phosphodiesterase enzyme. I have mentioned here, and it actually make the or actually reduce the CGMP. That is the main theory. Here increases of CAMP CAMP and here decreases is CGMP, and this occurs in case of rod cells by the photon, actually light. And here the beta 2 adrenergic receptor means epinephrine or adrenaline. You can say epinephrine or adrenaline hormone. This can be also some neurotransmitter. Adrenaline can act as a neurotransmitter. Then glucagon, serotonin or vasopressin, this can activate this pathway. G alpha I is activated by CGMBPD, actually activates, and this is actually sorry, G alpha, not G alpha, this is the G alpha T. It would be the G alpha T, and this is the G alpha I, because G alpha T means T for transducins. This transducin present in the rod cells, and this is the way. In case of G alpha I inhibitory protein, they actually inhibit the G alpha I inhibit the adenylyl cyclase enzyme, and the G beta gamma actually activate the potassium channel in the muscarinic acid, and how it works I will tell you later. So that's why, as it is inhibitor of adenylyl cyclase, it actually reduces the CAMP production. You can see here it is the inhibitor of this acetylcholine, so the amount of CAMP will be decreased in this signaling pathway, and as it is opens the potassium channel, so it will actually change the membrane potential also, change the membrane potentials. This is present in actually activated by muscarinic acetylcholine receptor, and also by alpha 2 adrenergic receptor. You must remember adrenergic receptor, you must remember this. There are different type of modes like beta 2, beta 1, then alpha 1 I have mentioned here. Okay, so yes, this is the alpha I, means inhibitory. The Q is activated by this way, that means alpha 1 adrenergic. So alpha 1 adrenergic receptor, this alpha 2 adrenergic receptor present in peripheral and central nervous system, peripheral or central and central nervous system. Okay, alpha 1 adrenergic receptor actually activates this G alpha Q, and that activates the PLC beta, phospholipase C beta, that actually activates the second messenger IP3 and DAG, and ultimately activate the PKC. Okay, so alpha 1 present in the smooth muscle, I have also here written here, smooth muscle cells of intestinal tract and skin kidney, so I am not writing here. Okay, and the G alpha O is present, that actually here endothelial cells, so I am not writing here endothelial cells, and this also activate the same pathway, PLC beta, activate the PLC beta, but the last product is here nitric oxide. Okay, for olfactory, olfactions, odorant is the odorant, that is an odorant receptor that present in the nose, that actually sends the odorant, different type of odorant, and this olfactory actually activate the G alpha, actually the G alpha is same thing, it actually acts as a stimulatory, so it activates the adenylyl cyclase enzyme and activates the CAMP second messenger, increase the second messenger. So these are all actually basic concept about the G protein coupled receptor. In this class I will show you how muscarinic acetylcholine receptor actually inhibit heart muscle contractions. Okay, how it occurs, how this pathway occur actually. So let's suppose here this is the plasma membrane and this is the heart muscles. At first you have to understand that this is the example of heart muscle pacemaker cells, the plasma membrane of heart muscle pacemaker cells, and this is the G protein coupled receptor. Okay, let's suppose. So when acetylcholine receptor bind or muscarine, this could be acetylcholine or mascarine. Mascarine means, I have mentioned in my previous class that it is actually a fungal alkaloid. When bind with this G protein coupled receptor, it changes the conformations. Then the G alpha I bind with this G alpha I, because I have mentioned that in case of muscarinic acid, G alpha I actually gets activated. This G alpha I actually, when there is no ligand, bound with the GDP, but when ligand bind like acetylcholine or muscarine, then it actually associate, this protein actually associate or touch with this GPCR, and this actually change the structure of the G alpha I so that it can exchange the GDP with the GTP. Now this becomes active. As it is in the name suggests that inhibitor, so it actually inhibit the adenylyl cyclase enzyme. So the transformation from ATP to CAMP actually reduce. Okay, so it is actually inhibitor that reduce, CAMP level reduce. Not only that, I have mentioned that the trimeric protein, the G alpha protein, associate with the other two subunits called beta and gamma. Togetherly actually they form a complex. This beta gamma actually play a major role in this case. They activate a receptor, a channel proteins called potassium channel. This potassium channels actually, when this beta gamma touch or attach with this potassium channel, it actually open the potassium channel. Potassium actually come out from the cells, and we know that when positive ions come out from the cell, it becomes what happens? It becomes depolarized. Sorry, it becomes hyperpolarized, because the negative portion of the inside of the cytoplasm increases. Depolarization means positive. The inner of this cell becomes positive depolarizations. In case of hyperpolarization, what happens? The inside of the cell becomes less negative. The normal membrane potential of any cells is 70 millivolt. Okay, when depolarization occurs, that means action potential occurs, the voltage changes into positive plus 30 millivolt. But when hyperpolarization occurs, the value becomes minus 90 millivolt or so, means the less than minus 70 millivolt. And when any cells become hyperpolarized, becomes less negative than the resting membrane potential. This is the resting membrane potential. When a cell becomes rest in conditions, they actually have a voltage inside that is negative minus 70 millivolt. But when it becomes hyperpolarized, that means when positive ions actually come out from the cell, it becomes hyperpolarized, become negative, more negative than the resting membrane potential. Then this cell cannot be stimulated. So that's why in case of muscarinic acetylcholine receptor, heart muscle pacemaker cells actually slow. The contraction becomes slow because it cannot be, contraction of the cell actually becomes slow because it cannot be actually activated, because it becomes hyperpolarized when acetylcholine or muscarinic binds with this receptor in the muscle cells. Okay, in my next classes I will show you how this process actually occur. Hope this video will help you. Thank you for watching, and always keep learning.